My dear students, hello. Myself, Dr. A. Jansari, Vice Principal, Associate Professor and Head, Department of Biochemistry, St. Kestimal Purwal College, County. The topic on which I am going to talk is a part of BSc Semester 6 syllabus. Yes, the topic is Shine-Dalgarno sequence. The Shine-Dalgarno sequence is associated with the correction of correct AUG codon for initiation of protein synthesis. Protein synthesis in prokaryotes begins with 30S pre-initiation complex that includes 30S ribosomal subunit, a mRNA molecule, charged tRNA with formulated methionine, 3 initiation factors IF1, IF2 and IF3, and a GTP molecule. It is a fact that the codon AUG acts both as a start codon as well as the codon for amino acid methionine. As we know that methionine occurs within the protein chains, there must be some signal in the base sequence of mRNA that identifies the correct AUG codon as the start codon. If the whole 30S pre-initiation complex is treated with the enzyme RNase that digest RNA only if it is free and single stranded. The portion which is bound to the ribosome is protected and the remaining free single stranded region is digested. This protected fragment can be isolated and its base sequence can be determined. One such protected fragment is shown below. In this diagram we can see there are two rows. The above rows is showing the base sequence of mRNA. The below row is showing the base sequence of 16S rRNA. The figure we will describe later on. This protected region of mRNA is usually 30 to 40 nucleotides long and always contains an AUG codon, that is the start codon. It also contains the codons for the initial amino acids of the protein adjacent to the AUG codon in the 3' direction. The AUG codon is usually 20 to 30 bases away from the 5' end, and just before this AUG codon, a sequence known as leader sequence is present. The most notable feature of this leader sequence is the presence of a sequence as follows. As you can see, this sequence is having large number of purines, so it is a purine rich sequence. This purine rich sequence is containing 4 to 7 bases and is present just before the codon AUG, the start codon. This sequence actually provides a binding site for the ribosome as well as defines the start codon and is called the Shine-Dalgarno sequence, named after the researchers who first recognizes its significance. Thus, the AUG codon, which is adjacent to this leader sequence in the 3' direction, will always act as start codon. The importance of this Shine-Dalgarno sequence becomes evident when the 3' end of the 16S rRNA of bacteria is examined. The region in the 16S rRNA contains the sequence as shown below. The sequence in the 3' region of the 3' region of the 16S RNA incidentally have the complementary basis with the Shine-Dalgarno sequence and can thus base paired with it as shown in the figure. This pairing brings the AUG codon to a start codon to a start site on the ribosome for initiating the protein synthesis. Thus, it is concluded that number 1, an intact 3' end of 16S rRNA is essential for initiation of protein synthesis. Number 2, certain ribosomal proteins involved in initiation are located in the ribosome near the 3' end of the 16S rRNA molecule. And number 3, antibiotics that inhibits initiation of protein synthesis are found bound to these ribosomal proteins next to the 3' end sequence of 16S rRNA. We will go back to the figure. So as it is shown in the figure, there are 2 rows. The above rows is that of mRNA molecule showing the base sequence there, and the below row is that of the 3' region of the 16S rRNA. So in the above row you can see the dotted 7 base sequence, which is nothing but it is the Shine-Dalgarno sequence, and the AUG codon which is adjacent to this Shine-Dalgarno sequence in the 3' direction is actually acts as the start codon. And in the second row, that is in the 3' end region of the 16S rRNA, incidentally it is having the complementary basis of the leader sequence, that is the Shine-Dalgarno sequence. So with this I conclude. Thanks for quite listening. Thank you very much.